Hey, it's Adriana from Dranalytics, and welcome to this introduction on NCBI's Sequence Read Archive. In this first part, we're going to be getting comfortable with navigating around the SRA website, and in the second part, we're going to be downloading data from the database. All right, so let's get started. By the end of this tutorial, you'll know how to locate a project or study listed in a paper, navigate around the SRA, and make sense of all the accession numbers you're going to find, the data types they hold, and their organizational hierarchy. So just to zoom out and give you an idea of what kind of sequencing repositories are available to you, there's one out of the US, which is the Sequence Read Archive and what we're going to be focusing on today, and that's from the NCBI. There's one from the European Bioinformatics Institute called the ENA, and then there's also one out of Japan, and that's the DDBJ Sequence Read Archive. If you click on the link that's included in the written component of this tutorial, you'll see that we're trying to download bacterial sequences from this paper on an organism called Burkholderia thailandensis. Scroll down all the way to the supplementary information, and there's a technical appendix which indicates what accession number we're going to be using to look for the data initially. That number starts with the letters SRP. So what I'm going to do is simply copy that number and pop it into NCBI's Sequence Read Archive. What this is going to do is drop us on to a website that gives us all of the information and accession numbers that are related to the study that was indicated in the paper. So we have some information on the sequence, the sequencing platform, who it was submitted by, something called the study, the sample, the library, and as well as some run data. So when we click on the study accession number, we find that it brings us to a page that's not really giving us a lot of information, but it's just telling us what this particular study is about. There can be multiple studies within a bioproject. This example is pretty simplified because it only has one. Starting at the very top of the organizational hierarchy is going to be the bioproject, and these accession numbers start with PRJN. So by definition, a bioproject is a collection of biological data that's related to a single initiative. So you can kind of think of it as a parent directory that's going to hold all of the metadata, all of the sequencing information, and any other related information that's related to the project. So we have a bioproject, then we have the study within that bioproject, and now within that study, we can have multiple experiments. So let's go back one page. And if we click on the experiment accession number, we're going to see that it brings us to pretty much the same page. You're going to see a lot of repetition here because the NCBI is going to try to present you with as much useful information as possible. So there's going to be a lot of crosstalk. This is going to be important later because it's going to give us information that's specific to the experiment design. So just a lot of sequencing data that might be helpful for your analysis later on. Underneath the experiment, you can also have, it's not really underneath, but beside, I guess, are the bioproject samples. Now the sample is just giving us information regarding the biological source of the genomic material used in the sequencing project. So in the paper that we're looking at, if we click on the sample accession number here, it's going to tell us the source of the genomic material, which was a forearm wound fluid. And so like there might be multiple studies in a bioproject and then multiple experiments within a study, there can also be multiple sources of biological samples. So now we're getting to the goal of what we're really looking for, which is the raw sequencing data, and that's going to be under the accession numbers that start with SRR. So if we go back to our summary page, the runs are going to be listed here, and you can click on them and that will bring you to a run browser. You can also click up here and that will bring you to a page where you can download accession numbers if there are multiple runs. In this case, there was only one run. Like I said, this is a really simple example. So we'll just click on that one run. And this is going to bring you to a page that's presenting you with the same information that we are looking at previously, just in a more visually appealing way, I suppose. So we have a bioproject here listed on the bottom, and then some information on the biosample. Up here is the experiment data. So we have some information on the library they used, the platform sequencing equipment, so an Illumina sequencer. The strategy was whole genome sequencing, the source of their material, the selection type, and the layout. And then the last one at the top here is a little bit of quality control data on the specific run that we clicked on. So as an aside, there may be a time when you encounter accession numbers that don't start with S, but they start with an E or a D. And this simply means that the data was initially submitted in the European Bioinformatics Institute for anything that starts with E, and then anything that starts with D would have been submitted through the DDBJ. And they're cross-listed on NCBI's SRA. So if we go back to this run browser, we see we have some metadata about our run. We have some experimental taxonomic analysis on the organism based on the submitted sequence. Here we can actually look at the read data. So it lists all of them, and they're separated here. Remember, we said that they were paired end. And then when we want to download the data, we click here, and we see that we actually need to download a separate piece of software to do this. So downloading data from the sequence read archive is not necessarily straightforward. And that's something that we're going to cover in the next tutorial. So I hope you were able to learn a little bit about NCBI's SRA. Thanks, and I'll see you in the next lesson.